Okay, thank you. In the name of God. In continue our previous lectures, today we are planning to start number 4, mutualism or symbiosis. Okay, thank you. Hope the best. Today we are planning to start mutualism or symbiosis. It is like previous one because the explanation is the same. Both populations benefit from the relationship. It is similarity with previous one. But the difference is here is obligatory. Did you remember in previous one? Synergism or proto-cooperation, both populations benefit from the relationship. Number 3, not obligatory. But here, there is obligation. Obligation. First, we go through historical example. According to the evolution of living organisms, in the after creation of the earth, the ancestors of yeasts and some bacteria came together. Yeasts and what? Some bacteria. Because those yeasts could do only fermentation and those bacteria could do respiration. So those bacteria penetrate yeast and live there. Yeast give them food and bacteria did respiration. You know respiration? Respiration is aerobic. Respiration means doing glycolysis, Krebs cycle, and electron transport chain. 1-glucose 36 ATP is produced. And after some time, yeasts understood bacteria as a good friend. And bacteria understood yeast as good friend. So they continue to live with each other permanently. Permanently. And as now, the grandfathers of mitochondria in our cells are those bacteria. And the remaining cells are those yeasts. So here, right now, if you take ribosome, if you take mitochondria from cells out, those mitochondria cannot continue life. And our cells without mitochondria cannot continue the life. So here, right now, mitochondria is beneficial for cell and cell is beneficial for bacteria, and this is obligation. This is obligation between both of them. Relationships of mutualism allow organisms to exist in a habitat that couldn't be occupied by either population alone. Even though the population may exist separately in other habitats. We have a theory of serial symbiosis. Some endosymbiotic relationships had key roles in the evolution of higher organisms. Now our cells, animal cells, fungi cells, there are cells with mitochondria. So mitochondria ancestors are bacteria. And after some time, their relationships now is obligatory. And on the other hand, some early yeasts, they go through symbiosis or mutualism with photosynthetic bacteria. So as you see inside plants, inside algae, there are some chloroplasts. The ancestors of chloroplasts are those photosynthetic bacteria. Photosynthetic bacteria. And now, one plant cell without chloroplast cannot continue life, and one chloroplast without plant cell cannot continue the life. So according to serial symbiosis, those population benefit from the relationship that it is obligatory. We cannot take them separate from each other. But nowadays, there are many other examples, not historically, like lichens. What are lichens? Some living things are present on rocks, are present on stones. We call them lichen. What are they? Lichen is the relationship between a photosynthetic organism like algae or cyanobacteria with a heterotroph with the name of fungi. So there are two in one. There are one unit containing two populations. One population is autotroph or primary producer. We call it phycobiont. And a heterotroph or a consumer or mycobiont. So in lichen, we have a phycobiont and a mycobiont together. Make a unit. Make a unit. What are they do for each other? Why fungi like algae and why algae like fungi? Why one producer should like a heterotroph like fungi? We understood why fungi like algae. Why? Because phycobionts, by use of light and carbon dioxide, produce organic compounds that utilized by mycobiont. So algae or cyanobacteria produce food for these heterotrophs. Okay, so 50% is okay. 50% we understood that heterotroph like autotroph, it is easy to accept it. But what do fungi for algae? Mycobiont provide a form of protection. Don't forget, they are living on a stone. And photosynthetic organisms and algae, cyanobacteria, are very sensitive to heat, sensitive to dryness, to many things. So fungi cover and protect it. Protect his friend. Another thing is that phycobiont algae or cyanobacteria for photosynthesis need light, water, and carbon dioxide, as well as minerals like phosphate, potassium, sodium, and the other minerals need For example, if one person wants to cook anything, first you should buy her those things need. If heterotroph, if fungi ask algae to produce food, first should give algae those minerals. Fungi produce acid, solubilize those minerals present in the rock, receive it, and give it to algae. So algae understand that this fungi is very good to have friendship with that. Algae give food to fungi. Fungi give minerals, give water, give protection for algae. So both of them are happy to live with each other. Okay? Microbiome provides a form of protection and mineral nutrient transport as well as growth factor for phycobionts. Algal and fungal members of the lichen form distinct layers that function as primitive tissues. See, when we look to microorganisms like bacteria, if we have, for example, E. coli, all E. coli do the same job. And whatever one can do, the other can do. Whatever one cannot do, the others cannot do. When we have algae, all cells do the same. When we have fungi, all cells do the same. And if one cell cannot do anything, the others cannot. But look at your body. You have cells, but different types of cells which they are in tissues. Your kidney tissue is different with liver tissue, is different with lung tissue, is different your brain tissues. In the same time, it is unity in diversity. You can see unity in diversity in your body. So many diverse cells are in your body, but they work in unit. They work in unit. So during the evolution, those microorganisms, those microbes understand they can come together and one population do something and the other population do the other things, which these things complete the unit. So lichen is a unit composed of two primitive tissue. One tissue is doing photosynthesis with production of food, and the other tissue, the other population, is responsible for protection and mineral, finding minerals. Okay? So it is the start of evolution. It is the start of formation units with diverse cells. Now we can see two different cells in one unit which complete each other. Phycobionts, the producer, the photosynthetic part, can be cyanobacteria, can be chlorophycophyta, or xanthophycophyta, nostoc, or green algae like Terboxia. According how much lichen can attach to the substrate, to the rock, to the stone, we can divide them in three groups. Group 1. In group 1, we call them crustose. Group 1, we call them crustose. Those lichens which adhere very closely to their substrate or stone, we call them, they are crustose. It is very difficult by finger you take them out from the rock. They are very closely attached to the rock. In the second group, which we call them folios, they are not so hard attaching to the rock, leafly in form and attached to substrate more loosely. And the third group, fruticose, very loosely. The loosest attachment is there, consists of a hollow upright stalk and are loosest attached to the substrate. So according how much is the strongest or weakness of attachment lichen to the rock, we can divide them into 3 groups. So now there is one question. Is the life on rock is easy? So if we can see in all rocks, we cannot find any microorganism or macroorganism to live there. So living on rock is not so easy, because we cannot find anything else than lichen there. So what is the benefit for lichens to live on rock? What is the benefit? The benefit is that they feel quite well because there is no competitors. No microorganisms come on stone to disturb them, to take their food. Live there is hard, but there is no disturbance. Clear? Do you get the point? Why do lichens prefer to live on rocks which the other microorganisms don't like it? Do you get the point? Understand? Okay. Very good. So lichens believe hard living is better to go to a situation with competition with many thousands groups of competitors. They don't like to compete with other microbes. They like to be in a place, silent, cozy, far from the others, and by hard living continue life without disturbance. Okay. Lichens are resistant to extremes of temperature and drying, so they can grow on rock surfaces. So low temperature, high dryness, many adverse effects, they are very resistant, which is present on the life on rock. Lichens produce organic acids that solubilize rock minerals, aiding their growth on rock. Yes, fungi do it. So till this part, we understood that phycobiont doing photosynthesis. What is photosynthesis? Photosynthesis is carbon fixation. What is the meaning of carbon fixation? It means carbon dioxide by help of light and water. And minerals can be changed to sugars. And sugars are used for energy production, ATP production. So lichens, because they have phycobiont, can do the carbon fixation. As well, some lichens can do nitrogen fixation. Nitrogen fixation. What is the meaning of nitrogen fixation? In carbon fixation, carbon dioxide in the atmosphere was used for sugar production. In nitrogen fixation, nitrogen in the atmosphere by some microbes and lichens can be used for amino acid production. Amino acids are used for protein synthesis, for enzyme synthesis, and so on. So cells without enzymes, without structural protein, cannot continue their life. Nitrogen source is very important. We cannot do nitrogen fixation, yes? We are dependent on plants and animals. We eat plants and animals to receive ready-made amino acids. And we use those amino acids for our enzyme production and structural protein production. So lichens, some lichens, as well as carbon fixation, can do nitrogen fixation also. In tundra soils, lichen pelticula containing nostoc provide major source of fixed nitrogen for biological community. So in these regions, lichens receive molecular nitrogen of atmosphere, use them, change them to the accessible forms, and fixed forms of nitrogen are leached from lichens during rainstorms and washed to the forest floor and available for forest vegetation and take by plant roots. So when you have a small yard, every year you should go to find and bring and use fertilizer for 2-3 plants which you have, 2-3 trees which you have. But in forest, nobody give them fertilizer. So naturally, most of these lichens give nitrogen source needed for plants to their roots. Even though we told that lichens are very resistant to changes of temperature and low water, but lichens are sensitive to industrial air pollutants. Lichens are sensitive to industrial air pollutant. So in cities and in industrialized places, you cannot find lichen. So if you go somewhere and you see there is lichen, it's a good place to have a good time there because it has a good climate. Because you are far from chemical toxic substances come from industrial factories. And in your cities, if you have a large number of cars and factories, never you can find any lichen. Why? Because lichens are very sensitive to industrial air pollutants and disappearing from industrialized areas in cities with polluted air. Because sulfur dioxide in atmosphere in these regions, in industrialized areas, sulfur dioxide in atmosphere is inhibitory and killing phycobiont. And phycobiont inhibition, we can see. When phycobiont is killed by sulfur dioxide, fungus are unable to survive alone, and fungus will go to die also. So, for example, if liver of one person not working, all the other tissues cannot continue life. If one person heart is not working, brain not working, liver not working, kidney not working, so now it is a unit. Lichen is a unit. If one part is not working, the other part stop work and cannot do its job after all. Okay, so the first example was lichen. In what? In mutualism or symbiosis. What happened? Algae help to fungi, and fungi help algae, and it is obligatory. If one stop working, the other is going to die. This relationship is obligatory. This unit only can be functional if both of them are working. If one is not working, the other stop life. Another example is endosymbionts of protozoa with algae. So now we have a unit and two diverse population, protozoa and algae. In previous one, we have fungi and algae. Here we have protozoa with algae. Protozoa with algae. Protozoa like Promysium, stellate, protozoan, Promysium. Inside Promysium, mostly we can see many algae with the name of chlorella. Because chlorella are alive in this unit and live inside Promysium, we call it endosymbiont. Don't forget, we are talking about symbiosis. In symbiosis, when one population is living inside another population, we call it endosymbiont. Endosymbionts. Okay. What Promysium do for chlorella and what do chlorella for Promysium? Algae is easy, yes? What algae do for Promysium? Algae produce food and oxygen. Finish. So Promysium is very happy. Promysium is happy. Some microbes live inside. Some algae lives inside and produce food and oxygen. What Promysium do in parallel to the algae? So many parasites, many predators are there for algae. When algae are inside Promysium, they are protected from invaders. So number one, protection. Number two, motility. What is the motility benefit for algae? What is the benefit of motility by protozoa for algae? protozoa go towards light. Because algae cannot go up, down, okay? But protozoa can go. When protozoa go to the direction of light, algae produce food. So Paramecium thinks it is in a correct way, but in a time which Paramecium going to dark, algae don't produce food and protozoa change the way of motility, change its way. The other thing, Paramecium during metabolism and using food during metabolism producing carbon dioxide, and carbon dioxide is what necessary for photosynthesis by chlorella. So chlorella like it. And as well, Paramecium produce growth factor. And growth factor facilitate growth of chlorella. So chlorella think Paramecium is a good friend, and Paramecium think chlorella is a good friend. And when in this unit, the diverse two populations are living together, both of them feels well, both of them feel very well. But the main thing, the main important thing is that Paramecium need more food. Paramecium is aerobic protozoa, and it can search food only in aerobic places. But those Paramecium which contain chlorella and can have extra oxygen can search not only aerobic areas, but also they can search food in anaerobic areas. So when Paramecium has a good friend like chlorella, can search food all over the habitats. There is no limitation for Paramecium to go different places. Paramecium going everywhere to find food, and everywhere algae is happy to receive many things from Paramecium. Note 1. Suppose you put this Paramecium containing chlorella in a flask, and you put the flask in dark place for long time. If for long time there is no light, there is no photosynthesis. When there is no photosynthesis, algae don't produce oxygen, don't produce sugar, don't produce food. So after some time, Paramecium thinks that it is not a good friend. Only using those things are present in Paramecium and without any production. So friendship is not one-directional. When it is one-directional, it is not friendship. So Paramecium digest and eat the chlorella. If for a long time don't show its friendship, if don't show oxygen production, if don't show sugar production, without light for a long time, protozoa digest the algal population. We have another endosymbionts. Similarly, if instead of chlorella, cyanobacteria, chlorella is eukaryote and cyanobacteria is a prokaryote. If cyanobacteria is replaced by chlorella, which occur both in freshwater and marine protozoa. We call this relationship the relationship between protozoa and cyanobacteria. We call this unit is cyanella. This unit is cyanella. It was example number 2. Now we are going to the example number 3. Endosymbionts of protozoa with bacteria. So previous one, protozoa with algae, producer, photosynthetic. But here, heterotroph and heterotroph. Both of them are heterotroph. Now we want to see which types of friendship can be here between a protozoa and with a non-photosynthetic bacteria. Some bacteria multiplying, they are alive and multiply in nucleus, others in the cytoplasm of some protozoa. For example, some protozoa have problem to produce hemine itself, need someone produce hemine for them. So some bacteria are present inside protozoa, so it is endosymbiont. Endosymbiont means friendship. One friend is inside another friend. One friend is inside another friend. There is a unit. One is there and the other is inside the first one. So bacteria inside protozoa provide hemine as well as some growth factor and give to protozoa, and protozoa do protection, give food and whatever bacteria need only because of this service. Because bacteria give service to protozoa. Protozoa is home, is house for bacteria and give bacteria whatever they need. Both of them are happy. Both of them are happy. The other example related to the protozoa and bacteria, both are heterotrophs, is related to the Paramecium aurelia and Caudibacter. As its name shows, Caudibacter is a bacterium, prokaryote, and Paramecium is eukaryotic, protozoan, eukaryotic, protozoan. Those groups of bacteria present in Paramecium come together and we call them kappa particle. So groups of Caudibacter as kappa particles are present in Paramecium aurelia. Accordingly, we can divide Paramecium aurelia in two groups. Group 1, killer. Group 2, sensitive. What is the difference between killer and sensitive? Killer Paramecium aurelia contain endosymbionts of Caudibacter. Sensitive Paramecium aurelia lack endosymbionts. So Paramecium aurelia in number 1 is endosymbiotic with Caudibacter, but in number 2 is not. Why are they killer? Why are they killer? Because Caudibacter present inside Paramecium aurelia can produce toxin, and use against the other competitors. And these genes of toxins are located on plasmids of Caudibacter. So if Paramecium aurelia contain Caudibacter and can produce toxin, kill the competitors, and we call it killer Paramecium aurelia. And if they don't have endosymbiotic relationship, so they cannot be successfully present in that habitat. Presence of endosymbionts give an advantage to killer strains in competition with sensitive or another groups for competition of food. Okay, 1 minute please. Again, we will start after 1 minute. Okay. Okay, we'll follow the lecture again. The last example. These examples are not all examples because I manage the lectures according to the syllabus and the time. So our time allow us to discuss this much. After the finish the lecture, you can go through the net, you can go through the different books, and you find the more beautiful examples and understand the way. Only the lecture is look like a brightness to show you the way. The lecture is not the complete way, only showing you the way, and you can have many more better and more valuable examples. Okay, last example is related to the endosymbionts of bacteria with lysogenic phages. As you know, phage or bacteriophage are those viruses can infect bacteria. Phage. Phage are those viruses can infect bacteria. Whatever we hear about viruses mostly are related to the medical virology. Medical virology is a course which explains which viruses can be pathogen for human. So accordingly, all the time you hear viruses are obligate intracellular parasites. What can you hear? Viruses are obligate intracellular parasites. Why? Because mostly 99% or 100%, whatever you read are related to the medical virology. But here, this example, there is no parasite. It is endosymbiont, because parasite is harmful for the other population. But here, virus is helpful. Virus is beneficial. What happened in this example. In this example, we can see some viruses are not lytic. Some viruses, if come to cell and immediately multiply and kill the cell, it is lytic cycle. But in some cases, when viruses come inside bacterial hosts, they are lysogenic. They have lysogenic cycle. What is the lysogenic cycle? In this cycle, when bacteriophage or bacterial virus inject its chromatin or its chromosome or its genome to the bacterium, this genome don't try multiplication, but it joins to the bacterial chromosome, and we call it prophage. What do we call it? We call it as prophage. What is the meaning of prophage? When a viral genome incorporate or join to the bacterial genome, and when we look to the bacterium, we see one chromosome. But this chromosome has two parts. One part is bacterial chromosome, and the other part is viral genome. So, which benefit bacterium has for virus and which benefit virus has for bacterium? Because it is endosymbiosis. Both should be friends. Virus here is not pathogen. Virus here is not enemy for bacterium. Virus is friend. What is the friendship? See, when bacterium multiply to 2 and chromosome of bacterial chromosome multiply to 2, we have 2 bacterium with 2 chromosome, each contain viral genome. When it is 4, 8, 16, 18, 36, 64, 128, whatever we can see the bacteria, all bacteria contain viral genome. So it is the benefit of bacterium for virus. During its multiplication, help virus dividing, but in parallel, not in lytic stage, not in lytic state. Okay, so it is benefit of bacterium for virus. What is the benefit of virus for bacteria? Some toxins genes are present in the genome of virus. So by presence the lysogenic phage, by presence of prophage, bacteria can produce toxin to attack to human, to have greater virulence factor. So virus make bacterium more powerful. So virus is helping bacterium, and when bacterium is powerful, help virus for dividing. Like what? Like Corynebacterium diphtheria. In Corynebacterium diphtheria, those one without prophage, very easily immune system suppress them and kill them. So those Corynebacterium diphtheria which are not infected by prophage, by virus. They are very weak, with weak virulence, and very easily are killed by immune system. But those Corynebacterium diphtheria with prophage, they are very strong. They are very powerful. They are toxin producer, and they can remain for a long time in our body, and sometimes maybe to produce serious infections and serious disease. Okay, 1 second. Okay. The other interactions between two populations we call as competition. Competition. What is the competition? Competition means two populations need food, and there is a limited same food they need. Same food they need. So they try to have it. Both populations try to have it. But don't forget, in competition, two populations never attack to each other and never produce toxin against each other. So how do competing? By fast eating, by trying fast growing, by trying fast multiplication. So when competition starts, when same food is present, but the present food is limited and is not sufficient for all members, and the population which fast receive food and fast grow and fast multiply is the winner, and the other population lose the game. And it is negative relationship. It is negative relationship between two populations, adversely affected the survival and growth. Competition occurs when two populations utilize the same resource, whether space or limiting nutrients such as carbon, nitrogen, phosphate, oxygen, growth factors, water, and so on, are limited. See, same resource. For example, there is a food table. You have a food table and two children. There is meat and fish on the food table. One of your child doesn't like meat and the other doesn't like fish. So what is the competition for? There is no any competition. It should same resource. For example, put one ice cream on the food table and two children. Then you can see very easily what happened during competition, because both of them like it. So it should be same resource, and should be limited. You put 100 ice cream on the food table and two children. What is the competition for? So each eat 1, 2, 3, 5, or 10. What's about them? Nothing. So they don't compete each other when food is more than they need. They should be same resource, and the same resource should be limited according space or limitation in the nutrients. And on the other hand, when we talk about the food, we are talking about regarding carbon, nitrogen, phosphate, oxygen, growth factor, water, and the others. If one is not present, for example, if phosphate is limited, nitrogen cannot be used, in a state of phosphate. If water is not sufficient, growth factor cannot use in a state of water. So all should be in one place. By use of all of them, populations can grow. Sometimes carbon source and nitrogen source are high. There is no competition for them, but phosphate is less. Sometimes competition is only for one item or for two items. Sometimes for all items, maybe competitions are present. Sometimes at home, all the things are too much. Only ice cream is limited. So competition is only for ice cream. There is no competition for the others. Sometimes father forgot to buy many things. Nothing is in the refrigerator, or very less are in the refrigerator. So competition is for all the things which they are limiting. Okay, in this part, we divide competition in two parts. During active growth condition, and in next page, during non-growth condition. What is the difference between these two? During active growth condition. During active growth condition. In this situation, all the things are okay. All environmental factors are okay. pH is okay, oxygen is okay, temperature is okay. All the things are good. Only one food is limited. Okay? All environmental factors are okay. Only one food is limited. In this situation, neither organisms attack to each other or secreted toxic substances. It is our rule for competition. But for example, if we have two paramecium with the names of Paramecium caudatum and Paramecium aurelia, both of them in a flask with their foods are bacterial prey. Paramecium caudatum wants to eat bacteria. Paramecium aurelia wants to eat bacteria, but it is limited. It is not sufficient. So they go for competition. After 16 days, only Paramecium aurelia survive. So after 16 days, there is no Paramecium caudatum. After 16 days, Paramecium caudatum is not there. Why? Because Paramecium aurelia has more rapid growth rate and outcompete Paramecium caudatum for available food. So Paramecium aurelia very fast finding food, very fast use it, very fast growing, very fast multiplying. So Paramecium aurelia is the winner population. And Paramecium caudatum lose the game. Even though they don't attack to each other, even though they don't produce toxin to each other, but the one with faster growth rate is the winner. Is the winner. Another example, Paramecium caudatum and Paramecium bursaria. Food is the same, bacteria. If bacteria as a food is limited, if bacteria as the food is limited, there is something here. They go to different side of the flask or habitat. They don't come close to each other. When one go to the left side, the other go to the right side, and they grow, so minimize competition. In this situation, competition is minimized. Survive and reach a stable equilibrium even when grows together. Even though they competed for the same food resource, the two populations occupy different regions of the flask. Ability to occupy different habitats provide a mechanism that minimized competition and prevented extinction of one of the species. So here, after 16 days, one is complete winner and the second is completely loser. But when they go to different sites and they don't come close to each other, winner and loser come for a very longer time. They can live together for a longer time with competition. So soon or late, one is the winner and the other is the loser. We call it competitive exclusion principle. According to this principle, one population during competition exclude or kill the other or remove the other population. One will win the competition and other will be eliminated on the ability to utilize a substrate more rapidly. And all these explanations are during active growth conditions. All environmental factors, pH, temperature, and salt concentration, all the things are okay. Only populations have one problem: limited food. So when they are in this situation, soon or late, one will be the complete winner and the other is the complete loser. And we tell this story as competitive exclusion principle. It is a principle which accordingly during competition, one population exclude, remove the other population. Okay, in a few minutes we finish this part. Example number 3, algae. Don't forget we are in competition part. Do algae compete with each other? They are photosynthetic. They can produce sugars. Yes, don't forget I told you that human and algae have similarity and differences according food. What is the differences between human and algae? Algae can produce sugar by using carbon dioxide, but human cannot. Should receive it from eating plants and animals. But what is the similarity? Both of human and algae have same similarity in food. Both of them receive minerals like phosphate, calcium, potassium, zinc. Yes, these things both of us we need. So algae have competition for minerals because there is no competition for carbon dioxide. In atmosphere, it is full of carbon dioxide. Never plants and algae going through competition for carbon dioxide. There are large number of carbon dioxide there. But mostly algae, plants, photosynthetic organisms going through competition for what? They go competing for phosphate for minerals. We have two algae with the names of Astrionella and Cyclotella. When phosphate as a nutrient is limited, Astrionella is the winner in competition. But if silicate is limited, Cyclotella is the winner. So here, like before, we see there are competition between two populations. But one thing we can understand, it is not so if there are two populations. In all cases, one is the winner, and in all cases, the other is the loser. No. In some cases, maybe one population A is the winner and B loser. In another situations, population B is the winner and A is the loser. If one student knows very well ecology, maybe another student knows advanced bacteriology better. We cannot say if one student is good in one course, must be winner in all courses. Okay? So there is competition in some cases. For phosphate, Astrionella is winner, and for silicate, Cyclotella is the winner. Or when sewage is running and going to the ocean or going to the sea, and near the beach is full of organic matters, and far from the beach is low substrate. When near the beach in the sea is high substrate concentration, E. coli is winner against Spirillum. But when we go far from the beach in the sea at low substrate concentration in marine, Spirillum is the winner against E. coli. And in the last part, all of these examples were related at the time, which all environmental factors were okay, were good. Only food was limited. In the last part, for terminating this section, environmental factors may be problematic. Like what? For example, there are not sufficient water. Here, the enzymatic power of microbe, like before, is not so important. It is not important which one can eat fast. Now, when there is no water, it is important which population is more resistant with dryness. If now the weather is hot, it is not important which has the capability of fast growing. Now it is important which population is more resistant to this hot climate. And the winner is the one which has more adaptation to that situation. Example, under condition of drought, population 1 can best tolerate and survive desiccation. And population 2 is sensitive to desiccation. So it is very easily can be understood that population 1 is the winner because they are better adapted and they are effective competitors under these conditions. Suppose when AC is working in the class, one student answered the questions very well. But when AC is not working, that student cannot think very well. And those students are winner, which can be more tolerated to the heat, to the hot situation, to the hot situation. It is what we should explain today. So next week, we go through the amensalism, If you remember in this lecture, I explained that in competition, there is no attack, there is no toxin production. In amensalism, it is a type of competition with toxin production. It is a type of competition with what? With toxin production. So next week we go through amensalism and we explain it. Is it any question? Okay, our lecture over. If it is any question, please ask me. Okay, thank you very much. Very good.